Okay, so welcome to this next video in the playlist on synaptic mechanisms. In this video, what we're going to do is we're going to talk about the role of a protein known as Rab3A in neurotransmission. So the title for this video is just this protein, Rab3A. Right, okay, so this is the setup for this video. We're going to look at the basic setup that happens in neurons. We're going to look at neurotransmission. We're not going to go through the details of it, we're just going to look at the broad picture of it. We're then going to see what Rab3A is and where it's present. We're then going to talk about an experiment which showed that Rab3A was in the vesicles. And finally, we're going to have a look at what happens if you knock out Rab3A. And I'm afraid, again, it is a protein where we don't really know that much about what it's doing in neurotransmission, but we do know it's important, as the knockout study will show you. Okay, right, so the broader picture then, if this is an axon terminal here, then we know that there are two stores of synaptic vesicles within an axon terminal. One is the readily releasable vesicle pool, which is this store of synaptic vesicles which are docked at the presynaptic membrane. So these are synaptic vesicles that are docked at the presynaptic membrane. So let me just colour them in with neurotransmitter inside them here. Okay, so these are the readily releasable vesicle pool, this store of vesicles that is docked at the presynaptic membranes. This is the readily releasable vesicle pool. And when an action potential arrives at the axon terminal, these synaptic vesicles that are in the readily releasable vesicle pool, those are going to be the first vesicles that will fuse with the presynaptic membrane and will release their neurotransmitter contents into the synapse. Now, then we also have another store, which is basically the reserve pool of vesicles. Now, these are vesicles that are held back further inside the axon terminal, and they are attached basically to the actin cytoskeleton of the axon terminal. So here are actin filaments of the that make up the cytoskeleton of this axon terminal. So what do I mean by that? Well, how do cells maintain their shape? Why do they stay in this shape? Why does this axon terminal stay in this shape? Is it the cell membrane? Absolutely not. The cell membrane is just a gooey mush. It's completely fluidly moldable, okay? So it's not the cell membrane that keeps the axon terminal in this shape. Instead, inside the cytoplasm of the axon, what you have is a very, very fine meshwork of proteins all over the place, basically, which is keeping the axon terminal in this shape. And one of those key proteins, those cytoskeletal proteins that hold up the shape of the cell, is a protein known as actin. Okay, and these are actin filaments. Okay, so actin itself is a tiny little globular protein. Actin filaments are polymers of actin. So if we draw an actin monomer as just a little circle, which we'll colour in green, then an actin filament is basically loads of these actin monomers all joined together in a polymer like so. So you'll polymerize the actin monomers together to make a larger actin filament, like so. Okay, so that's what an actin filament is. And basically, you can attach synaptic vesicles to the actin cytoskeleton. So here is our synaptic vesicles which are attached to the actin cytoskeleton. So the vesicles that are held behind the presynaptic membrane in this so-called reserve pool. So this is the reserve vesicle pool. Okay, so this is where we're going to get our vesicles from when we release all of these ones that are in the readily releasable vesicle pool. Okay, so these vesicles, these reserve synaptic vesicles, they are attached to the actin cytoskeleton by proteins known as synapsins. So here in pink, this is a synapsin protein, And this larger blob that I've drawn here, this is the synaptic vesicle. So here are the neurotransmitters within the synaptic vesicle. Okay, so this protein holding the synaptic vesicle to the actin filament, this is known as a synapsin. Right, okay, these synaptic vesicles are held to the actin cytoskeleton by these synapsins, and synapsins have a site which binds onto the actin filament on one side, and then on the other side they'll have another binding site which binds onto the lipid of the phospholipid bilayer of the synaptic vesicle here. So this is a synaptic vesicle which is going to be held in the reserve vesicle pool. Right, now, when an action potential comes along down the axon and arrives at the axon terminal, it's going to cause very, very quickly the release of the vesicles from the readily releasable vesicle pool. So all of this neurotransmitter will be released. Now, after that, we're not going to have any vesicles left over docked at the membrane. So, if another action potential comes, what happens? Well, what has to happen is we have to replace these vesicles that are docked at the plasma membrane. So, what happens is we liberate vesicles from the reserve pool, and they have to go and dock at the membrane. So we replace the vesicles which are docked in the readily releasable vesicle pool via taking vesicles from the reserve vesicle pool and docking them at the membrane. Okay, so that's the big picture of the process of regenerating the readily releasable vesicle pool, so that when another action potential comes, you have vesicles docked at the membrane ready to fuse. Okay, right, now let's look at this protein Rab3A. So, in order to tell you how they found that Rab3A was in vesicles, basically, I want to tell you about synaptosomes. Okay, so let me tell you about what a synaptosome is. So, a synaptosome. Right, so a synaptosome then, what you do is you take neural tissue. You take tissue containing neurons. So let's say we have here a neuron. Okay, so these are the dendrites. This is the axon here, and here's the axon terminal right at the end. So this is our neuron. What we're going to do is mildly homogenize it. So we're going to put our nervous tissue through mild homogenization. Homogenization. This is a challenge on the old spelling. Homogenization. Okay. Right, and what we're going to get is these neurons aren't going to remain intact. They're going to be chopped up into loads of little pieces, but we're not going to completely mash them up. We're going to do a mild homogenization. And if you do a mild homogenization, what can end up happening is in the homogenate, which you get at the end, what you can end up with is these axon terminals here. So let me highlight it. You can end up with an axon terminal on its own, basically, not attached to a neuron, but on its own. So you can end up with like a you can end up with an axon terminal on its own. So here's this axon terminal. It's had the rest of the axon chopped off it. That was chopped off it by the homogenizer. But if we only do mild homogenization, then the axon terminal might just happen to remain intact. Now, when it happens that it has its axon axon chopped off it, what happens is it fuses back up. The membrane fuses back up. So you end up with this structure which is merely an axon terminal, which will have all the machinery of vesicles docked at its membrane, so here are these readily releasable vesicle pores. So basically, it's just an axon terminal. This is what is known as a synaptosome. So basically, it's these things that you can get out of homogenizing neural tissue mildly, and they basically form from axon terminals that have been chopped off neurons. Okay, now, these synaptosomes are very, very useful for doing experiments with regarding neurotransmission, because they still will release neurotransmitter in response to depolarization. So what you can do is you can now depolarize the membrane of one of these synaptosomes, and in the presence of extracellular calcium, of course, and you can do experiments into the synaptic vesicles. And through this, what they were able to find is, firstly, in the membrane of these synaptic vesicles, there is a protein known as Rab3. Okay, so let's draw this protein here. So we'll draw it like this. Okay, so this protein, it represents, well, this drawing that I've put here represents the protein Rab3A. Sorry, Rab3A. And Rab3A basically is within the membrane of these synaptic vesicles. So here is Rab3A. Now, what is its function? Well, basically, through experiments on synaptosomes, what they were able to find was that Rab3A drops out of the membrane of the synaptic vesicles when you stimulate this synaptosome. So let me say that again. We're going to stimulate depolarization of the membrane of this synaptosome, and in the presence of calcium, that's very important. If it's not in the presence of calcium, then nothing happens, because fusion is reliant upon the depolarization being transformed into a calcium signal. Okay, so if you depolarize it in the presence of extracellular calcium, what's going to happen is you're going to get calcium going into the synaptosome, and it causes somehow Rab3A to drop out of the membrane of the synaptic vesicles. So these vesicles that are going to fuse with the presynaptic membrane when the calcium comes in, the Rab3A drops out of those vesicles before they fuse. Okay, so why does this happen? Well, basically, in order to gain some more insight into what Rab3A's function in life is, we can do knockout studies. So what we're going to do is we're going to take some experimental animal, we're going to knock out Rab3A, and we're going to erase it. So we're going to perform a knockout experiment. Now these are pretty commonplace now. They're usual protocol if you want to find out what a protein does and whether it's important. You can find a mouse, or often we do it now, well, you can do it on mice. Another species that you often do it in is C. elegans, Caenorhabditis elegans. So let me just go over some of the experimental animals you can do knockout experiments in. So firstly and foremostly, the ones that we've been doing it in for a long time are Drosophila, which is a type of fruit fly. Okay, and these have the advantage that they reproduce very, very quickly. They grow very, very quickly. So you know, you can do the experiment quickly. And also the fact that when you're doing genetic experiments, obviously people get very emotional about genetic engineering of animals. And flies are one of those animals that it's, you know, you need a more hardcore anti-vivisectionist to oppose an experiment on a fly, basically. Whereas if you were doing it in monkeys, obviously you'd have a lot larger opposition, basically. Okay, so Drosophila is an animal that we often do genetic experiments on. You can also do it on Caenorhabditis, and I've learned how to say this, Caenorhabditis elegans, which is often just abbreviated to C. elegans to avoid having to say Caenorhabditis. But this effectively is a worm. It's a worm, and again, it's easy to grow, and again, it meets little opposition from anti-vivisectionists. If you have a bigger license from the Home Office, you can also do it on mice, basically. You can do knockout experiments on mice. What they did is they do these knockout experiments on mice usually nowadays, and what happened is the mouse was absolutely fine. If you knock out both genes for Rab3A, the mouse is fine, apparently fine. It's completely happy, completely normal. So they had to search pretty hard to find anything that was wrong with this mouse, and eventually what they did find, they found one little change. Now let me explain this, because this is quite a complicated and subtle change. So, if I put in two neurons, let's say this is our presynaptic neuron, so this is our axon terminal of our presynaptic neuron, and then here is the dendritic spine of our postsynaptic neuron, which the presynaptic neuron is synapsing onto. So this is the postsynaptic neuron, This is the presynaptic neuron. Right, okay, so what we can do is we can continually stimulate this presynaptic neuron. So I can put my electrodes in here and I can continuously stimulate this presynaptic neuron so that it fires action potential after action potential after action potential after action potential, basically. Okay, so you continually fire action potentials down this neuron, and let's say we do it at a frequency of, I think they do it usually at around 14 Hertz. Okay, now what does that mean? That means that in a second, so let's say this represents a second, 1 second, that you will fire, you will cause the the presynaptic neuron to fire 14 times. So you'll fire 14 times, and I'm not going to be able to fit 14 of them into there. But the point is that you stimulate it 14 times in a second, and they're evenly distributed. So you do it every 1/14th of a second. So you stimulate, wait 1/14th of a second, stimulate again, wait 1/14th of a second, stimulate again, etc. So it's firing action potentials continuously. Okay, now what you also do is you record the effect of the action potential here. You record the excitatory postsynaptic current. Excitatory postsynaptic current. Okay, so let me explain what this means. Synaptic current. Now, excitatory postsynaptic current is often abbreviated to EPSC. Okay, and what this means basically is you measure how much sodium ions are coming through into this dendritic spine because of the neurotransmitter. So let's say we are releasing glutamate, which is the most common neurotransmitter in the central nervous system. So we're releasing glutamate. Glutamate is going to diffuse across the synaptic cleft, and it's going to act on glutamate receptors on the postsynaptic neuron, which may well be AMPA receptors, let's say. AMPA receptors are going to open and allow sodium into the cell. You will measure that movement of positive charge into the cell, and basically what you'll do is you'll plot this against time. So here against time, and then we plot the excitatory postsynaptic current, which is just how many sodium ions, how much positive charge, remember each sodium ion carries one positive charge, how much positive charge is coming through the membrane. So what you'll see is something like, it will go up when you first stimulate it, and then it will fall back down as the neurotransmitter goes down, and then it will go up again, and again. So basically what you see is every time you stimulate this presynaptic neuron, the postsynaptic neuron is going to get some excitatory postsynaptic current. And I just want to redo this graph because I don't like that. Okay, so let's say it's plotted here instead. So what you'll say is every time you stimulate it, you'll get a certain amount of excitatory postsynaptic current. So let's say the first time you stimulate it, you get some amount up here. And what you'll find is that as you continue to stimulate it, the amount of excitatory postsynaptic current you get drops off like this. That happens in normal neurons. Now, why is this happening? Why, if we continually stimulate it like this, is the amount of excitatory postsynaptic current we're getting per stimulation of the presynaptic neuron, why is it going down? So let me say that again. We're stimulating this presynaptic neuron continuously, and what we're finding is the amount that each stimulation of the presynaptic neuron, the amount of stimulation produced in the postsynaptic neuron for that action potential in the presynaptic neuron is going down, basically. Why? Well, it's because effectively the amount of neurotransmitter is going down, because it's using up its vesicles, basically, and gradually the amount of vesicles that will be docked at this membrane when the next action potential arrives is going to get fewer and fewer and fewer, and that's why the excitatory postsynaptic current induced by each of these presynaptic pulses is going down. Okay, now what you find in Rab3A knockouts is that if you repeat this experiment, the rapidity with which this effect is seen is much quicker, so it goes down maybe something like this instead. So this depression in the stimulation of the postsynaptic neuron that you see per action potential after you've repeatedly applied action potentials, this occurs quicker in Rab3A knockouts, and this is known as post-Titanic depression. Okay, so the post-Titanic depression is greater in Rab3A knockouts. Right, now what does that mean? That means that the recruitment of vesicles in the reserve vesicle pool is somehow impaired in Rab3A knockouts. So remember, every time we fire an action potential, we're having to recruit vesicles from the reserve vesicle pool to the readily releasable vesicle pool in order to replace those that we've released. So if the post-Titanic depression is happening quicker, then that means that we're not managing to recruit these replacement vesicles as well. So that suggests that Rab3A is involved basically in the recruitment of the reserve vesicles and allowing them to dock at the membrane, and that might be why it drops off when you actually fuse the vesicles, because the Rab3A needs to be recycled and go and help new vesicles to be recruited to the plasma membrane or to the readily releasable vesicle pool. So we think Rab3A may well be very important in the docking process.